In today's video, I'm going to be talking about DNA replication and how this process occurs. This is a follow-on video from my last video on DNA and RNA, which I've left a link to in the description, but I would highly recommend watching that first, as there are a lot of key concepts that you will need to understand in order for today's video to make sense. So, the current theory of DNA replication is a process called semi-conservative replication. And when this occurs, what happens is that our DNA double helix will unravel, and this allows an enzyme called DNA helicase to bind to it. And as helicase runs over the DNA molecule, what it does is it breaks the hydrogen bonds that occur between the base pairs on each DNA strand. And this will leave the DNA as two single polynucleotide strands. Now, these strands are known as the original strands, and they are able to act as a template for new DNA strands to form. So we essentially go from having two polynucleotide strands to having four polynucleotide strands. And this occurs because as helicase comes and unravels and breaks these hydrogen bonds, it leaves the DNA bases exposed, and this allows free floating nucleotides that just occur within the cell to be attracted to these exposed bases, and they form complementary base pairs. So when you think back to the last video, cytosine always bonds with guanine and adenine always bonds with thymine. Now, as this occurs, another enzyme gets involved, and this enzyme is DNA polymerase. So as well as hydrogen bonds occurring between the base pairs, the sugar phosphate backbone in DNA needs to be formed, and this occurs by condensation reactions, and DNA polymerase catalyzes these reactions. So we think back to the last video, in a nucleotide, the sugar from one nucleotide forms a phosphodiester bond with the phosphate group on another nucleotide, and this allows our sugar phosphate backbone to occur. And as I've already said, hydrogen bonds have also formed between our base pairs on the original strand of DNA and our newly formed strand of DNA. So when this process is complete, we should have two new DNA molecules that have replicated from this original one DNA molecule, and each molecule will contain one strand of new DNA and one strand of original DNA. You can see this on my diagram that our original DNA strand is in blue and our new DNA strand is in red. So now let's talk a little bit more about DNA polymerase. On the right hand side of the screen, I've got this rather complicated looking diagram, but if we break it down, we can see that our DNA double helix has unwound to form our two polynucleotide strands, and we can see DNA polymerase is represented by this yellow block. We can also see we've got a red arrow which shows the direction that DNA polymerase is moving in. So DNA strands have different ends, and the ends are different because they have a different structure. So each DNA strand has what's known as a 5' end and a 3' end. Now if you visualize a DNA molecule, if we think about the top end of it, we've got the top part of our two DNA strands. One of these strands will be a 5' end and the other will be a 3' end. And this is because DNA is what's known as an anti-parallel molecule. So one strand will run with its 5' end at the top, and it will run in a 5' to 3' direction, so its 3' end will be at the bottom, and the other strand will have its 3' end at the top and its 5' end at the bottom. So DNA polymerase is complementary to the 3' end of the DNA strand. So again, visualizing our DNA molecule, if DNA polymerase were to bind to the molecule, you'd get one DNA polymerase that binds to the top of the DNA molecule, to that 3' end at the top, and you'd get another DNA polymerase that binds to the other strand, but at the bottom of the molecule, where its 3' end is located. And because enzymes are only able to bind to the 3' end, they will add new nucleotides into our new DNA strand in a 5' to 3' direction. And this is because DNA polymerase is always moving in a 3' to 5' direction. And because the double helix is anti-parallel, the new strand is always being made in a 5' to 3' direction. This is quite a confusing concept to get your head around. It's something I spent a really long time trying to understand. But go back, listen to this example again, look at explanations in textbooks, explanations on the internet, other videos, and hopefully it will start to make sense. But I am aware it is quite a confusing concept and one that's actually quite difficult to explain. Now, the DNA helix, as it's replicating, will have what's known as a leading strand and a lagging strand. Now, the leading strand is the one where our original template strand is running in a 3' to 5' direction. And this is our leading strand because DNA polymerase can bind straight away to that 3' end on our original DNA strand, and it can just move down in the 3' to 5' direction, making our new strand in its 5' to 3' direction without any difficulties. Now, in our lagging strand, the exposed end of our original DNA strand will be the 5' end, which is not complementary to polymerase's active site. So instead, what will happen is that in the unwound portion of the DNA helix, another enzyme will come in and it will break the sugar phosphate backbone, therefore exposing a 3' end of DNA. And what DNA polymerase will do is it will bind to this new exposed 3' end, and it will create the new strand of DNA by binding to this new 3' end and synthesizing our new strand in its 5' to 3' direction. And then once this has occurred, an enzyme called DNA ligase will come along and it will fix this break in the sugar phosphate backbone, so it doesn't remain broken forever. It gets joined back together again, and we will end up with 2 DNA double helices at the end. So on to the final part of today's video. We're going to be talking about something called the Messelson-Stahl experiment. So once Watson and Crick discovered their model of the DNA double helix, scientists then went on to try and discover how DNA replicated. And they came up with 2 theories, and these were known as conservative replication and semi-conservative replication. So conservative replication is a process where DNA will replicate. We will get 2 new DNA molecules forming, so 2 double helices, and one of these helices will comprise of the 2 original strands of DNA, so the 2 original strands from our original DNA molecule, and the other helix will be made up of 2 new strands of DNA. So the original DNA molecule is conserved completely, hence the name conservative replication. A semi-conservative replication is the process that I have just described, but DNA will replicate, 2 new double helices will form, and each will contain 1 strand from the original DNA molecule and 1 new strand. So the Messelson-Stahl experiment investigated how DNA replication occurred in bacteria, and they proved that replication was semi-conservative. And then other scientists went on to repeat this experiment with the DNA from other living organisms. And we have now got a universal method to say that semi-conservative replication occurs in all living organisms. So if I go through the steps of the Messelson-Stahl experiment, I will then draw some diagrams afterwards so that you can visually see the results of their experiment. But what Messelson and Stahl did was they took 2 samples of bacteria, and these were grown on a nutrient broth. One of these broths contains something called light nitrogen, and the other contains something called heavy nitrogen. Now, light nitrogen is our normal isotope of nitrogen. So nitrogen is an atom with 7 protons and 7 neutrons, and this is known as nitrogen 14 because it has a mass number of 14. Now, heavy nitrogen has, again, 7 protons, but this time it has 8 neutrons, so its mass is 15 instead of 14. And as bacteria reproduce, they take up the nitrogen from the broth and they incorporate this into their DNA nucleotides. So DNA samples were then taken from each batch. So a sample was taken from the bacteria that was growing in the light nitrogen broth, and another sample was taken from the DNA grown on the heavy nitrogen broth, and these were spun in a centrifuge, and 2 bands of DNA formed. So the heavy DNA was lower down in the test tube than the light DNA, because centrifugation works by spinning a sample of a material at a very fast speed, and the heavy elements of the material get spun out and land at the bottom of the tube, and things that are lighter will rise and they will settle nearer the top of the tube. So the bacteria that was grown in the heavy nitrogen were then put in a light nitrogen broth and left for another round of replication, and then that sample was centrifuged. So in Messelson and Stahl's experiment, they had their 2 dishes that they were allowing their bacterial DNA to replicate on, and one of these had our light nitrogen broth, so our 14N nitrogen in the broth, and the other contained heavy nitrogen, so our 15N nitrogen. What they found, so they took a sample of the bacterial DNA and they centrifuged it on what's known as a cesium chloride gradient. And what this does is that as they centrifuge the mixture, these the molecules or the components within the mixture will separate out according to their density. Now, the DNA that was growing in the light nitrogen broth was incorporating this 14N nitrogen into the bacterial DNA, and so overall it had a lighter density. And what they found in their test tubes that they were centrifuging in was that DNA taken from the light nitrogen broth formed a band quite high on their gradient, so near the top of the test tube. Now, with the bacterial DNA sample from the heavy nitrogen broth, so that had 15N nitrogen incorporated into its DNA, they found that the DNA band formed near to the bottom of the test tube, and this is because the density of this DNA was much heavier. Now, if conservative replication were true, when they then took this 15N DNA sample and transferred it onto a 14N sample, they would have expected that after another round of DNA replication, their test tube and their results would look something like this, and there would be a heavy band and a light band. And this is because if we think back to our example of conservative replication, we know that the original DNA molecule is conserved completely, meaning that the DNA that contains our heavy nitrogen will be conserved completely and will form a completely separate band, and that our new DNA molecule will contain only 14N nitrogen and will therefore be of a lighter density and form a band near the top of the test tube. Now, they actually discovered that this wasn't true at all, and what they found instead was, give me a second just to erase this test tube. What they discovered instead is that after a round of another round of DNA replication. So like I said, they've taken their DNA that's been replicating in this heavy nitrogen, they've transferred it to light nitrogen for another round of replication. And what they discovered was that a band formed in the middle of the test tube. So this band had an intermediate density, which could only mean that it contained one strand of DNA that contained only light nitrogen and one strand of DNA that contained heavy nitrogen. So what they theorized was that the heavy nitrogen molecules, when they were placed in this broth, they separated out into two separate strands, and the new strands that formed only contained 14N nitrogen, but that each molecule contained one strand with nitrogen of these different densities. And so their process, they deduced that semi-conservative replication was the process by which DNA underwent replication. So let's recap everything that we've covered today. We have learnt that DNA helicase breaks the hydrogen bonds between DNA base pairs, and that each original strand acts as a template for new DNA strands to form. We've learnt that complementary base pairs pair A with T and C with G, or adenine with thymine and cytosine with guanine. We've learnt that condensation reactions join nucleotides in the new strands and hydrogen bonds form between base pairs. And this is catalysed by DNA polymerase, which joins new nucleotides in our new strand in a 5' to 3' direction. We've learnt that the Messelson-Stahl experiment proved that DNA replication was semi-conservative and that centrifugation of their DNA mixture showed an intermediate band of DNA, so with an intermediate density, meaning it contained one strand of DNA with heavy nitrogen and one strand of DNA with light nitrogen. So now, as always, the best thing is to go away and recap this topic to make sure you understand it fully and then complete the following test questions. So number 1, what is the difference between conservative and semi-conservative replication? Describe the process of semi-conservative replication. In what direction does DNA polymerase form new DNA strands and why? Describe, with diagrams, the experiment performed by Messelson and Stahl to prove DNA replication is semi-conservative. And what bands of DNA would we expect with centrifugation of Messelson and Stahl's sample if conservative replication had occurred? That's all for today. Thank you for listening.